Good afternoon, Sam. Thank you so much for joining me and connecting to talk about your upcoming session at the conference in December. I first wanted to ask if you could please provide a little bit of your background, and then what led you to your interest in dementia research.
Yeah, so I'm a statistician. That is my background. Although I have some statistical genetics in there. So I've been in the pharmaceutical industry since I started in 2003. Um, been in different aspects of it. But what led to me being in dementia research in particular is that I connected with Suzanne Hendricks over 10 years ago, and she sucked me into her world, and we've been working pretty closely ever since.
Almost 20 years have passed between the approval of memantine in 2003 and the recent influx in the new Alzheimer's disease treatments that have been approved. So, based on your meta analysis, what was the biggest foundational barrier in the clinical trial methodology that created this void for promising drugs?
This is a really complex question because there are lots of factors that led to that giant gap. One of them just being that we kind of transitioned. Memantine, of course, is a symptomatic treatment, and the recent approvals are disease-modifying treatments or disease-targeting treatments. And so there has been a shift in the focus in trying to understand the disease and then address the fundamental characteristics of the disease to hopefully push towards something that could be a cure, or at least total disease modification, something that lasts. Where things like memantine and glantamine and donepezil, they all address specific symptoms and can give people a boost, but that boost will just go away as soon as it's out of their system. And so that would be one of the big shifts as far as what led to us going from memantine to aducanumab and donanemab and lecanemab. But what caused that gap besides that are just a number of different issues. So one of the issues that caused that gap is that because we're shifting from symptomatic treatments to disease-modifying treatments, we need to start thinking about how we measure them differently. Those symptomatic treatments can give you that great big boost, and in some ways that may seem like they are better treatments because the boost is so large. But over time, being able to modify the disease will give you benefits that will outstrip anything you could get from these symptomatic treatment effects. What that means is that over the short duration of a trial, that treatment might not look as promising as one of those symptomatic treatments. So, like I said, this is a complex question, but one of the key things is that we have treated disease-modifying treatments like we treated symptomatic treatments, which looked for large effects, maybe big effects on two outcomes that could be co-primaries. But because they're smaller treatment effects, and because you have to actually have disease progression in order to see disease-modifying effects, the effects that we see are smaller. And because we were stuck in a paradigm that was looking for symptomatic treatments, we ended up with looking for our effect sizes to be much bigger right from the beginning. And those were things that could have been measured by multiple primary outcomes, but that is a recipe for failure. In fact, it's a recipe that led to a 99.7% failure rate over that 20 years.
So, your research identified that there were 13 treatments that had demonstrated the evidence of efficacy as strong as or stronger than some medications that have been approved today. So, what do you feel was the most surprising treatment that showed signs of efficacy that was abandoned?
So one of the most surprising was choline phosphorate. And the thing that was surprising about that was the size of the effect in the studies. When we looked at all of the outcomes together, all of the evidence for it just had a massive treatment effect, and has been abandoned for a couple reasons. One being that it's not as lucrative for pharmaceutical companies to pursue this particular treatment because they wouldn't be able to have a large margin on it. But another one that was surprising are things like ginkgo and the boswellic acids because they are supplements. And that people have given up on them. But if, again, as we look at all of the evidence together, that those treatment effect sizes are actually big enough that that they should not be dismissed.
So what modifications to the trial design do you feel like could have resulted in a different determination for those specific treatments?
Yeah, so these ones actually are also more along the lines of symptomatic treatments, and so you'd think that just following the same paradigm would have led to a similar result. But ultimately, when we're looking at Alzheimer's disease and other complex neurodegenerative diseases or complex diseases in general, we have to understand that the outcomes that we are measuring might be too narrow. They may be measuring only a specific aspect of it, and that can be very successful, or it can be less successful because we are measuring just a slightly wrong part of the disease with that outcome. And so I tend to think of these sometimes as like investment portfolios for any savvy investor out there. One knows that that the safest route is to diversify your portfolio, and in this case, that doesn't mean doing co-primary endpoints. What it means is taking all of those different outcomes and putting them together into one outcome. The best way to do that, if we have narrowed in on a specific segment of the disease, severity of the disease, the best way to do that is through a composite score that has been specifically designed to align with disease progression, so that when we actually modify the disease, or when we are altering even through symptomatic disease, the specific areas that are covered by a composite, we have the best chance of seeing an effect. So that would be one thing that I would definitely encourage people to do differently, is to try and bring all of those outcomes into a single outcome, instead of assuming that just because you're modifying the disease, you are equally likely you have the same probability of hitting on one outcome as you do on more than one outcome. So that would be the biggest change that I would encourage people to do. There's lots of small things though. That's one of the reasons why we exist, because the disease is so hard. Allowing additional variability is not an option. And so even just designing your study from the get-go to have your database align with your CRF, which aligns with your data sets, the data set formats that you're using, which aligns with your table listing figures. If you can get all of those things to be in sync, you're gonna keep all of your data as clean as possible. So that's one thing. Another thing that we would recommend is making sure that your analysis lines up with your treatment effect, with the known issues. So little things like making sure you're including proper covariates, or handling post baseline initiation of acetylcholinesterase inhibitors. Just lots of little things like that. All of those things add up to a big failure if you aren't taking care of them.
What about, Sam, like gender differences? Is there value in creating a large enough subpopulation cohorts of male and female to tease and identify those differences, or do you feel like it's not necessary?
So I don't know that it's as necessary to focus on that. It becomes, the smaller your sample size, the bigger
Right.
that is going to be an issue. But by the time you get to a phase 3 trial, usually you're in Alzheimer's disease, the sample sizes are just by necessity big enough that the gender differences aren't going to be as important. Historically, the gender split has favored females, but as we move earlier in disease, it's going to shift towards males. Although what's interesting about that is even as it shifts towards males, it never it hasn't so far ended up favoring males.
In your abstract, you highlighted that there's an overemphasis on protecting against false positives while neglecting false negatives. So can you briefly explain what that trade-off means and why it is damaging for patients and caregivers waiting for the new treatments to come out?
Well, let me just start by talking about the evidence that we have overprotected our type 1 error, our false positives. So the evidence for that, first of all, is that 20-year gap we talked about earlier. That's part of the evidence anyway, because with what was in a few different sources cited as a 99.7% failure rate in Alzheimer's disease studies. One thing that we sometimes say is that we are owed some type 1 error. I mean, if we're using a 0.05 alpha, then we should have a 2.5% type 1 error on these studies, and the failure rate, even if they were all bad, would be 97.5. But I happen to know that they haven't all been bad. And so our failure rate should be something below 97.5%, and it definitely hasn't been. So that's some evidence of the type 1 error. One of the causes of that type 1 error that we've seen, and maybe more evidence that regulators are maybe a little too concerned in protecting against false positives, is the fact that we have been and still are encouraged in some settings to use co-primary endpoints. Just by their nature, a co-primary endpoint that is not between two endpoints that are perfectly correlated is going to overprotect type 1 error. It's going to discourage false positives in a way that is too conservative, that is going to cause good treatments to fail, it just by design. So we should not be using things that increase our type 1 error or decrease our type 1 error to a point that is below what has traditionally been accepted by regulators. So that 0.05, which is a 0.025 type 1 error rate for treatments that are not effective. But that affects your false negatives. So it causes us to increase our false negative rate. Therefore, we're not protecting the population from bad treatments at this point. We're protecting them from good treatments. And we should be doing everything that is necessary to get good treatments to patients. And anything that we're doing that prevents a good treatment from getting to a patient is by definition hurting the population. And we just haven't been focused on that enough. So one of the changes that I would advocate is if regulators could look more at risk-benefit analysis as they're evaluating drugs. For instance, if you have a very clean safety profile, the bar for approving that treatment should be lower than something that has a high has a high safety signal. And therefore we can push these things out, and by getting treatments to market, or in other words, by getting treatments to patients, we can start finding the things that work through larger phase 4 studies and just through post-marketing studies in general. Sometimes with these complex and difficult diseases, we need to get to a solution through iteration and not through one giant home run. I don't know. I believe that a solution to Alzheimer's disease isn't going to come through one home run. It's going to come through the iterative process of getting more and more effective treatments to market.
Some treatments, and you even noted, in the supplement category, there were some, they failed because they lacked that clear pathway forward for regulatory approval. So what should sponsors and researchers be focusing on earlier in the development to ensure those promising treatments don't hit that roadblock?
Yeah, so supplements are hard. Supplements and nutraceuticals are hard, specifically in the United States. So, for instance, there is one of the treatments that we really like is Suvinade, and Suvinade has been approved in Europe. It has been approved in Great Britain. It was approved in Australia. It is not approved in the United States, and the reason for that is it's a nutraceutical. It's like a supplemental type drink, and there isn't a clear path for approval. Even though they have run the studies, they have a clear demonstration of efficacy. Because there's not a clear pathway, it's a little harder to be able to approve these sorts of things. So. One thing that I would say for those looking to get a supplement approved in the United States is one of the first things we have to do is change the system a little bit. The other more interesting thing that could be done, and I will put this out there, not tongue in cheek, because it's a real avenue, but knowing that it is less than ideal. One thing that has been done, and I'm not going to say what the specific cases are, but it has been done, is to combine a supplement with a known drug. And the drug doesn't even have to be effective, but because you've done that, you've now created something that can be reviewed by regulators in the normal way. It might expose patients to safety signal, safety issues, adverse events that they would not otherwise be exposed to, but then they wouldn't also have the access through the same avenues to this treatment. And so there's trade-offs, and I would encourage, say, lawmakers and regulators to consider those trade-offs and look for ways to make it so that we can have approved supplements by the FDA that are approved for specific indications in the same way that small molecules and large molecules and devices are approved.
Your methodology also involved the global statistic test to combine the cognitive, the functional, and the global outcomes. So why is relying on a single isolated outcome metric inherently risky and prone to random chance in complex diseases like Alzheimer's?
Okay, so let me use one case that a lot of Alzheimer's disease researchers will be very familiar with, which is aducanumab. In aducanumab, they had a single primary outcome as their main outcome. It's well known. It's the CDR sum of boxes. This outcome, because there are a few different reasons why it created the controversy, and let just as a reminder, the controversy is that there were two studies. They were two identically designed studies. One is called Engage and one was called Emerge. Engage started a little bit before Emerge, and so they had more subjects that were represented in their interim analysis. In their interim analysis, they had a stopping rule that allowed them to stop for futility. They met that threshold and they stopped the study. And then as the data came in, the results showed that specifically in Emerge for the CDR sum of boxes, there was a positive treatment effect and it was significant. So there's a couple reasons for that. One of those reasons is a statistical one, which is that if you stop for futility, your treatment effect is of necessity deflated. It has been deflated, and it would be more deflated in the study that had a greater representation in that interim analysis, the Engage study. And that shows up in the fact that the CDR sum of boxes was not significant in Engage, but it was in Emerge. The second takeaway from this is that the CDR sum of boxes as a single outcome is more subject to variability because it is not measuring as much of the disease as would be if you had a composite or if you had a global statistical test that is taking into account multiple domains together. Hopefully, all major domains of the disease and measuring them at one time. This is again been seen when you look at the other two monoclonal antibodies that have received approval in recent years, lecanemab and donanemab. Both of those in their phase 2 study were had their CDR sum of boxes, the one primary outcome that the FDA has seemed willing to use for approval. Both of them, if they had to use the CDR sum of boxes as their outcome that they were making decisions on, both of them failed in their phase 2. However, thankfully, both of those studies, the Trailblazer study for donanemab and the Band 2401 study for lecanemab, both of them are using a composite outcome. With donanemab, it was the IDRIS, and with lecanemab, it was ADCOMS. Both of those composites are broader measures, and therefore A, more stable, and B, more likely to pick up disease modification. So because they used those as their primary outcomes in their phase 2 studies, they were able to move on with confidence into a phase 3 study. And thankfully, by the time they ran their phase 3 study, their sample sizes were large enough that that CDR sum of boxes actually hit for both of those. And so they had no trouble getting approval based on the CDR sum of boxes with a large sample size. But for many of the Alzheimer's disease researchers and sponsors out there, they don't have the funds in a phase 2 study to use phase 3 size sample sizes. And so if they aren't smart in the outcomes that they choose and make sure that those outcomes are broad enough in what they are measuring, then they are likely going to run into a situation where they are going to fail, even if they have an effective treatment.
So your conclusion states that every promising treatment failure that should have been a success moves the field away from what may be a critical component to the treatment of Alzheimer's disease. So if some of those 13 abandoned treatments you evaluated had been brought to market, how do you feel the landscape of Alzheimer's care would look differently today?
So for one thing, some of the failed treatments were actually disease modifying type treatments. So including like gantenerumab, which is still being investigated, but other studies as well. And so when we have failed treatments, we've talked earlier about the fact that we are likely to need every bit of incremental change that we can get. What happens when you get failure in life in general? When we fail, we get that negative feedback and we start looking in other directions. And we don't know that if we had just stayed researching certain things for 5 more years, what could have happened. And having, some of those successes would have encouraged some of the promising routes that were being investigated earlier to continue to receive interest and funding. So we don't know what treatments could have been, pursued if some of these that should have been approved had gotten approved. In fact, I believe that philosophers have shown that when we fail, we make more bad decisions than when we have some positive feedback included. So we start grasping at lots of different things. And so a few years ago, we had there was a panel at CTAD or AAIC, and they were asking, it was after some of the successes with the donanemab and lecanemab, and they were asking what they're most excited about in the field. And so John Sims of Eli Lilly said that he was excited about the virtuous cycle that had been created by these successes. And that virtuous cycle, he specifically referred to was the fact that we have some successful treatments, which means that we need to have to have better biomarkers. And so more research was then established in the biomarker community. And now we have more biomarkers available that can better identify, Alzheimer's disease subjects in general, but also the specific severity of the diseases, and then also looking at how the biomarkers themselves might change. And so by not having more successful treatments earlier in the process, we missed out on some of the virtuous cycle, because then those biomarkers can lead to more opportunities for better treatments to pop up, and it would lift the whole field together. By making bad decisions with study designs, bad decisions with outcomes, bad decisions with analysis, we missed out on many treatments that should have come to market, and an earlier access to this virtuous cycle that will lead to better treatments and more precision medicine, and all of the many benefits that come from a few successes.
If I'm a researcher, a clinician, or a sponsor, we're each going to have our own lens. So, what actionable lessons do you think I would pick up from your upcoming poster session at the Clinical Trials in Alzheimer's Disease Conference?
So I would hope that a researcher would look and notice some of the treatments that failed, and say, oh, maybe this is there's a promising something promising along this type of treatment that can be used. If I'm a sponsor, I guess kind of the same sort of thing. But sponsors specifically would have the responsibility to make sure that they're not following the same design issues that that led to this. That they would have a little courage in choosing their outcomes, and not just go the safest route, especially in phase 2 studies. But I would say even in phase 3 studies, one of the greatest mixed results for me in recent history was the fact that donanemab with Eli Lilly succeeded on both iADRS and on CDR sum of boxes, which is great, and exactly the result that I hoped for, except for one thing. Their primary outcome was not CDR sum of boxes. Their primary outcome was iADRS, a composite score. But as soon as they hit on CDR sum of boxes, they could emphasize CDR sum of boxes, because that they knew that the CDR sum of boxes had found favor with regulators in the past. I would have, in some ways, it would have helped the field as much to have them hit only on iADRS, so that they would have to get the FDA to weigh in on the acceptability of a composite score like iADRS, and either approve or not approve based on iADRS alone, because by making that happen, it would have led to a greater number of trials that hopefully would be allowed to use better outcomes as their primary outcome, so that we would have a better chance of seeing these successes. So that's what I want to see from sponsors. From clinicians, well, what's interesting is I was in Korea not too long ago, and one of the treatments that they assign to patients as soon as they are diagnosed with Alzheimer's disease is choline alphosphorate. But also interesting about that is the fact that the government is now requiring them to have more definitive clinical studies to demonstrate the efficacy of choline alphosphorate. And I think a lot of the clinicians are thinking that it's not that they might not get success on that trial, although I suspect that they will. And so I think clinicians should know that there may be more treatments out there that are safe and effective. Specifically, they might look at some of these things like the ginkgo biloba and boswellic acids, and I'm afraid I don't remember the third one right now, but look at some of these supplements and understand that they were at least as effective as some of the treatments that are being prescribed to patients right now.